Then 14 online.
Hmm.
Yeah, 3 physical, 14 online. You need a name?
Yeah. Who are the physical ones?
Okay. Physical ones. Oh, I send you the screenshot.
Oh, is it just
What is it?
just tell me the
Chloe.
very cool.
Yes, correct. And then
Okay.
Bofang and Sarah Lim.
Sarah Lim. Yes.
Yeah.
Oh, there are two Jingpofang. Okay, sure. Okay, thank you.
Yeah.
Yep.
Okay. 加油加油.
Hello, do I call you Ariel or do I call you Ayman?
Ayman. I you can call me Ayman, yeah.
Ayman, hi Ayman. What school are you from?
St. Joseph's.
St. Joseph's, awesome. You just send out the link to the for the worksheet, And who is the full stop? I need you to put your name there, please. Any problems accessing the worksheet?
Nope, I can access it.
Awesome. And hello, full stop. Hello, full stop. I need you to change your names, display names so I can know who you are. And hello, Kaylin. Kaylin, hello. Can you see and hear me? Kaylin and Ray and Hannah, hello. Love it if you all can let me know that you can see and hear me. I assume that's Phoebe. Hi. Kaylin, M, and Ray. It'll be more helpful if you can put your like name there instead of just the letter M. And then I have Grace. Hello, Grace. Well as well. Hello. Hi, Ray. Appreciate it. Eh? Ray? What do you mean? How do you know that? Kaylin, Megan, and Ray. Hi, hi. Ray has spoken, so I know Ray is here. Megan and Kaylin, hello. You here with me? Yes, Megan is here. So Kaylin. All right, so I hope you all are holding the worksheet I am sharing on screen, and let's get this started. Okay, so before we jump right in, we're gonna look at a quick introduction first. Kaylin, Megan, and Ray, have you attended any of our free revision lessons before? Megan has. Ray and Kaylin, what about the two of you? Kaylin has as well. Okay, so y'all will find this slide familiar. Just gonna go through it very quickly then. These few slides are all the same for all the tutors here. So maybe I'll just jump right into the one that is me. So this is a little bit about me. I've been teaching for more than 10 years. I love the sciences, especially biology. So, more about me outside the classroom as well. I just love being an outdoors and I love nature. And I'm actually also a climbing instructor, and I've taught a couple of classes here and there. So if you do want to find a spot to get into after your exams, hit me up. All right, so we are going to be looking at a few questions for these three selected topics. So let's jump right in. Okay, so we are looking at these three topics of molecular genetics, inheritance, and infectious diseases in humans. Kaylin, Megan, Ray, and our new friend here, H. Are we tested for these three topics for your end of years? Megan is. Okay, so let's get started. So as you can see for these three topics, right? Two of them have always been in the syllabus, and infectious diseases in humans is the one that is absolutely new. So I don't really have a lot of reference with regards to like school question stuff. But I have tried my best to come up with questions that I think are pretty much safe to say that likely will come up testing on our understanding of the requirements of the topic lah. We're gonna jump right in and do the questions together as a form of revision. We're not going to be looking at notes. So please feel free to refer to your own school notes as well as we are looking at the questions together. What I will do is not just look at the questions and answers. We're gonna dive deeper and see how we can take this opportunity to revise. So immediately you are looking at a gene and we're looking for the complementary strand. So when we say the word gene, we are thinking of DNA. And then when we think of complementary in DNA, we are going to be thinking of our four bases. So we have to recall to ourselves ATCG. Okay, all right. So adenine is complementary to thymine, cytosine to guanine. Right, we need to remember this immediately. Very, very straightforward. Must firstly immediately remember. All right, but I've left a space in between those letters because I would like to draw a couple of lines inside them. Kaylin, if you would be so kind as to join me with your microphone so that we can have a little bit of sounds here, not just my voice. Kaylin, can you tell me what kind of lines do I want to draw in between these few letters, Kaylin?
Um, is it covalent bond? Or I don't know.
Kaylin, are you the Kaylin that I know?
Yeah.
Fine, Kaylin. Okay. These nucleotides are going to be complementary. What holds the complementary bonds, complementary bases together? It is not going to be covalent. It is a much weaker one. Any idea?
Um, ion. No, I don't know.
Ionic bonds are quite strong as well, isn't it? Thank you for thinking about this, Kaylin. And yes, Megan to the rescue. Wonderful. They are hydrogen bonds. Megan, then can I have you help me? Adenine and thymine, how many hydrogen bonds? And then cytosine and guanine, how many hydrogen bonds?
A and T is two hydrogen and G and C is three hydrogen bonds.
Wonderful, right? So thank you, Megan. Right. So this actually also allows us to think about this further. Hey, the cytosine and guanine pairing actually confers greater strength to the entire DNA molecule, right? Because A and T are bound by two, while C and G are bound by three. All right. So therefore, the CG pairing will confer greater strength to the overall molecule. All right. So once you have this in mind, it's very straightforward. We're looking for just a guanine nucleotide. So we're looking for how many guanines are there. We therefore have to look at the complementary one, which is cytosine. So if there are 40 cytosine nucleotides on this strand, there will be 40 guanines on the complementary strand, and our answer is A. Very straightforward. Okay, right. So moving on over here, we are looking at an experiment, right, to produce designer milk, which contains specific antibodies. Question shows us a diagram of the first three stages of this experiment. We're looking for transgenic cells. Transgenic cells. Trans, we can break this word up. Trans, we're looking at different. And then for genic, we're looking at, well, genes and. So transgenic cells, different genes. Transgenic cells are therefore cells that contain genes from different sources, from different organisms. Right, so over here we have three stages. All of them have cells. We have a cell here, we have a cell here, we have a cell here. So of these three cells, which one or ones contain genes from more than one organism? That is what we're looking for. All right. Ray, do you want to help us over here, Ray? Which of these three cells contain genes from different organisms? Ray, we can see that this cell came from a cow and it was inserted with DNA from a human cell. So this cell will therefore contain DNA from here and its own. So stage one, we have one already. Right, Ray? So think about it and tell me, is it going to be just, well, man, I made the question too easy. We only have one stage one over here. This one is also a transgenic cell, right? So you can see that the nucleus was removed, so it's only going to be this, and I take this nucleus, put it in over here to form this thing. And if this nucleus is transgenic, this nucleus will therefore be transgenic as well. And our answer is A again. I have Johans, H and Shay joining us. So make sure you have the worksheet that I shared earlier on or in the Telegram chat. But if not, here it is again. Once more in the Zoom chat. Okay, let's move on. Question three, you have Escherichia coli. So, I believe that we definitely have encountered scientific names before. So it's kind of like a two-part name. So Escherichia coli. When we are writing it down, we must always make sure we underline them. So it is good for us to be familiar with the full name of this very, very, very common bacteria. You will also commonly see it written as E. coli, right? So it's a truncation of the word Escherichia. So, correct sequence, this one very basic understanding, right? We need to know what are the sequence of these stages. They may not be the full sequence, right? So there may be gaps in between, but we must obviously know that we must begin with you must first know what you're working with. If I want to make insulin, I need to know the gene that makes the insulin, right? Okay, so number two must begin with that. And I made the question too easy again, right? Because we only have one over here. And simply put, we're looking at cutting the gene out. Let's do some revision over here then. When I cut the gene, Johans, would you like to switch on your microphone and help me answer this, please? What do we use to cut genes in genetic engineering? Johans? Johans? Hello, hello. Yeah, maybe Joanne's not here. So thank you again, Megan. We're looking at the restriction enzyme. Megan, do you have to know the names of any particular restriction enzyme in school? Don't need, huh? Okay, so yes, thank you. All right, restriction enzymes are used to cut uh genetic material up, be it DNA or RNA. And then we're gonna transform the bacteria with the recombinant plasmid. So this is our second learning point as well. Recombinant plasmid means that it was recombined with something else. How do we recombine genetic material then? So this is our next enzyme that is very important in this topic here. Shane, you want to help me with this, Shane? Do you know what enzyme I'm looking for? After using restriction enzymes to cut the gene of interest and the plasmid up, I need to use this other enzyme to put the pieces together. What is the name of that enzyme? Shane. Shane, are you there? Looks like I'll just be working with Megan and Kaylin then. Thank you, Megan. Once more, look at didn't like it. Yes. This is an important point. We're going to be working with this idea again later on in the open-ended questions. All right. So once we have transformed them with the and into a recombinant plasmid, I'm going to put it into a cell. They're going to grow, and then I will extract it out. Answer is B. Fairly straightforward. Okay. All right. So here is an even easier question here, making sure we can identify the different parts in our DNA molecule. Kaylin, before answering the question here, pick one, A, B, C, D, Kaylin. Identify one of them, please. Okay, Kaylin, wonderful. Yes, so you have found the answer. This is our nucleotide. Okay, so we're looking at just this. What about A, C, or D? Can you identify any one of them? Yes, orange circle thingy is the phosphate group. Okay, so this thing is the nitrogenous bases. And then C, Megan, you help me over here. What does C refer to? Well done, deoxyribose. Oop, let me spell that properly. Okay, deoxyribose sugar over here. So you can see when I put A, C, and this thing together, they form B, which is the nucleotide. Question is asking for this. So what then do we call this thing in D, which is essentially the nucleotide minus the phosphate group? This is actually a nucleoside. All right, so nucleotide with a T versus a nucleoside with an S, right? The T is with the phosphate group. Okay, so knowing that we are looking at different things here, but obviously we need to remember the nucleotide, which is of more importance because that is what is the individual units that makes up the DNA molecule. Okay, so this is what we're looking at here in our DNA ladder, and the dashed lines in between represent the hydrogen bonds between the complementary bases. Okay. Okay, moving on to a little bit about inheritance already. So looking at what Lucy and Jordan are doing, heterozygous. Oh no, Katie left. Heterozygous. Okay, so Ray, Shane, H, Johans, last chance. Do y'all want to participate with me? If not, I'm just gonna ask Megan already. Can someone tell me what heterozygous refers to? Yes, so H and Johans are both here. Thank you for speaking. Right, so we're looking at heterozygous, meaning the alleles are going to be different. Right, so yes, both of you are correct. So they have different. So you can just choose any letter here. Johans went with this. So if both parents are heterozygous, what will their children be? Fairly straightforward. We're just gonna do a very quick cross. All right, just do a cross out. Okay, both of them are gonna be. What am I doing? For my quick workings in MCQs, I just do Punnett square, fairly straightforward. And And I will just see this is my homozygous dominant, heterozygous, and homozygous recessive. And we are looking at this sentence over here. Albinism is due to the expression of the recessive allele. So this is where we have to recall, hey, what does it mean to be a recessive allele? Recessive alleles are only going to be expressed in the absence of dominant alleles. And this is our only instance here. So only this child will be an albino, while the other three genotypes will not result in albinism. So first and foremost, the moment we see recessive allele, there's not going to be any half phenotypes. Recessive dominant are used to describe complete dominance, the allelic interactions. So there's not going to be any half. We're only going to be looking at this kind of half ideas. We are talking about co-dominant or incomplete dominant alleles. Right? So definitely this one is going to be out. Okay, the child with the recessive phenotype is what we have found over here. Heterozygous, this one shows me homozygous recessive. Again, therefore, this is going to be out. So between 3 and 4, hey, keep in mind the phrasing I've chosen to write over here. 50% of their children being heterozygous. So, of the 4, I have 1 homozygous dominant, homozygous recessive, and then these 2 are heterozygous. Meaning, of the 4 total genotypes, 2 of them are heterozygous, which is a 50%. And hence, this one is correct. Looking at our 4th statement over here, 66 heterozygous. But a very, very important point to look at is there non-albino children. So we have to look at within these 3, how many are going to be heterozygous? Within 3, there are 2 heterozygous, so there is a 2/3, and hence 66%. So we're looking at 3 and 4. Oh, I did not put the options properly. All right, okay, so please help me to change that. Maybe we can have this as our 3 and 4. Why did I not change it? Okay, right. Okay, so simply put, just make sure we're looking at 3 and 4 as true statements. 3 and 4 are true statements, okay? Did I not make the change? Okay. So okay, right. So let's move on now. We are looking at the red hat trait. We can see that question did not give us the information on which trait is dominant or recessive. We are just given the inheritance pattern. Right? We have the non-red hat and we have the red hat. Question looking for definitely heterozygous. Definitely heterozygous. So a quick tip I always share with my students here is that if you are presented with this kind of family trees and you are not provided with the information of who is dominant and who is recessive, right? Always look for whether or not something was hidden. Hidden meaning what? If you look at number 10, number 10's red hair was hidden in the parents, right? Okay, so that's what we are thinking of. It will not be this was hidden because it was present here, and it will not be this was hidden because I'm present here. Whereas this is hidden because these two don't have. So this tells me that red hair, therefore, is hidden and recessive. Once we can identify the recessive phenotype, we can go ahead and label them. So I always go on to find out all my homozygous recessive genotypes first. And then I think to myself, hey, I have 2 of them, 1 from each parent. So this one is 1 recessive and don't have red hair, so heterozygous. All of the white shapes have at least 1 dominant allele. If the child has 2, means came from 1 each, hence this way. And over here has to have come from this because cannot have come from 3. and therefore the other allele would have came from 3 heterozygous. This one must have come from this because the dominant allele cannot have come from this, and hence this is from here, this from here. I do not have information here yet. So therefore, this one, question mark. And who else? Who else is a question mark? This one. Why is this a question mark? This uppercase can have came from here or here. And this one could also be the other one as well. All right, so individuals 4 and 9 may be homozygous dominant or heterozygous. Question looking for definitely heterozygous, and hence 1, 6, 7, 8. Which do I need help in physics? Well, I hope you are looking for some help in biology as well. Welcome here. So let's move on. Question 7. The nightingale is a bird, and for those who do take literature, you may be. Oh no, that's mockingbird. Okay, never mind then. Okay, nightingale is an insectivore. Insectivore, just like carnivore means meat eater. Herbivore means plant eater. Insectivore is an insect eater. So the ancestral species of this bird had a less pointed beak. Over time, and when I say time, it's like thousands of years and up to millions, they have evolved to have a more pointed beak, right? So beginning with less, now is more. Why so? Catch small insects. So we're looking at why would this evolutionary change have occurred? Okay, so keeping in mind, you need to have an advantage when you are talking about something being passed down from generation to generation and being kept in the population as well. So we're looking for advantages. So we're looking at catching small insects means better able to find food. Okay, better able to find food. So are there other do the other sentences give me this advantage as well? They develop more pointed as they search for insects in the cracks, which means that today the bird got a less pointed beak. Then after a few months, because he kept searching for food, that bird's beak got sharper. That is not how evolutionary changes occur. You must keep that in mind. Evolution is about the population, not about the individual. Okay, so that's not going to be true. Now, C says more pointed beaks are more likely to seek out mates with less pointed beaks. Definitely not correct. Okay, so keep that in mind. Wait, can I just check first? Megan, your handout, right? Your worksheet. Are the options the same as mine, the order? Okay, okay, very good. Right. Thanks for answer. Right. So C says that female birds with more pointed beaks are more likely to seek out mates with less pointed. So this would actually mean that more pointed mixed with less pointed. Then you have more of like an overall even distribution. And that's not what we're looking at, right? Because now all of the nightingales have more pointed beaks already. Okay. And finally, we are saying that sexual selection occurs in the direction of seeing more pointed beaks as more attractive. While it may be the case, we do not have that information from this question because the only information given to us is catching small insects. So that is a food thing, and our answer is A. Okay. So keeping in mind what I shared with you earlier on about how evolution occurs over thousands, if not millions of years, here we're looking at just 20 years the effect of applying pesticide over here on houseflies. Okay. Question A is very similar to question 7 in terms of what I want, what I hope that you all can learn from is we are looking at the few resistant houseflies. Hey, why will there be resistant houseflies in the first place? All right, this one we are reminding ourselves that there is natural variation within the population, right? So natural variation within the population, so some will be naturally resistant already, or even just more resistant and not entirely immune, right? So they can reproduce more successfully, while the less resistant ones will just die and not be able to reproduce, right? So the very fact that the more resistant houseflies can survive, they can reproduce, which then reminds us again about inheritance. You're passing down genes, right? So whatever genes, whatever alleles allow these houseflies to be a little bit more resistant to the pesticide would be passed down to the offspring as well, which will cause this resistance, this idea, this frequency of resistance between the houseflies to increase over the years because of sexual reproduction, and therefore whatever gene, whatever allele that resulted in the resistance increase in frequency, meaning more of them are now present. Okay, more of them are now present. All right, so A is our answer. B is wrong because an increasing proportion mutated. Well, mutation will occur, yes, but it may not necessarily result in resistance, right? Mutation can result in even no change at all in the phenotype, even though there's a genotypic change. Okay, repeated exposure to the pesticides cause the houseflies to become more resistant. That is desensitization of the individual. If you were to use your right hand to slap your left hand repeatedly, after some time, it will feel less painful, even though you are slapping at the same intensity. So your pain receptors in your skin are now less sensitive. That happens to you. If you stop slapping now, tomorrow start slapping again, it will feel painful again. All right, so that happens to the individual over short periods of time. This graph shows us the percentage survival of the species. Did someone want to ask a question or something? Okay, all right. There is survival of the species, so it's not going to be the individual. And last but not least, the allele for resistance mutated from the recessive to the dominant form. Right? If A mutated to B, A is no longer present. So this phrasing of the sentence is incorrect already, which then leaves us with the first correct answer of A. Okay, moving on to will not result in antibiotic resistance in bacteria. Okay, so this one is our infectious diseases in humans already. will not result in resistance. So again, thinking about how resistance can evolve to can how something can evolve to have a resistance, they must be able to survive and reproduce. Very important. Must be able to survive and reproduce. Okay. So which of these statements therefore will not result in the bacteria being able to survive and reproduce? I think this is fairly straightforward. We are looking at wrong colors. This one. When you do not complete a course of antibiotics, the antibiotics are meant to kill off those bacteria. And if you do not fully consume the proportion assigned to you by the doctor, you may leave some bacteria remaining in your body, which then like, wow, suddenly nothing killing me. I can reproduce. Okay, so that will result in those being able to reproduce faster and better simply because they survived the antibiotics. Okay, the other three, wearing a face mask when indoors, right? So what does masking up do? What does masking up do? We are looking at Megan, any idea what does this prevent the spread of? Oh, Ray and Vishwain, what about you? Can we have your help here, please? Ray and Vishwain? If two of you don't need this, then not gonna waste time on this already. Ray, when you say you don't know the answer to this question, but it's in your syllabus, or you don't know if this thing is in your syllabus. Which one has left? Ray, hello, Ray. So need or not? Well then, let's skip it. Let's skip this to all these questions then. Don't waste time for you, Okay, so just making sure that molecular genetics and inheritance, H, Megan, and Ray, in your syllabus, molecular genetics and inheritance. Okay, H says yes, Megan says yes. Ray, what about you? Awesome, let's go on then. We have someone joining us now. Okay, so we're looking at genetic engineering. So we need to be very familiar with the steps necessary over here. Right? So we are taking out cells from the calf stomach. So calf is a baby cow, if you did not know that. Okay. We're gonna take out those DNA, and I use restriction enzyme A to cut those DNA out. That's just the blue segments. Okay. At the same time, maybe another researcher will be doing this, taking Escherichia coli. I'm gonna extract the plasmid first, and I'm gonna cut open the plasmid with restriction enzyme B. I'll put them all together, allowing them to base pair to form the recombinant plasmid. I'll transform it by forcing the plasmid in, and this allows me to get the transgenic bacteria, which we talked about earlier on about what transgenic refers to. And then at this point, I'll let it grow, and once it's at a large enough concentration, I will remove all the bacteria and extract the protein that the bacteria has made for me. Once Johnny has showed the professor this diagram, we are looking at one error, one missing component. One error and one missing component. Okay, maybe I let you all look at the question first. H, you are absolutely correct. Okay, you're absolutely correct. Well done, well done. But at this point, I just realized that this is the slightly older version of the worksheet that I wanted to share with you. So can you all actually download the worksheet over here, please? Okay. Got slight changes for this one. Oh, we've lost Ray. Okay, H and Megan, can you all let me know once you have downloaded the new worksheet? Got slight difference. Eh? Can't open. Why cannot open? Megan, can you also, you got any problems also? Can eh? H, you try again. See, can or not? Oh, H, any problems? Oh no. What about you, Megan? Can do you see the difference, this one? So the previous one using is the one that I was doing this morning. Then afternoon I made some edits. So it's like that. Right. So page, then you can see the difference here. That's why just now this one got A part 1 and 2 everything. Okay, then the MCQs also, the order of the answers got changed, right? So when y'all see the answer key in the future, it refers to the new worksheet as well. Okay. Okay, so H answered just now what was the error and the missing component and the missing step, right? So this one fairly straightforward. This one, the error is in the different restriction enzymes, and the missing component, right, is over here. We are missing DNA ligase, right? Okay. No, let's just hide this. Okay. All right. So we have to identify and explain the error. So identify the error. Didn't use the same restriction enzymes. Why is there an error? What would this result in? What would this result in? Come. But only got two of you only, so just whoever just say. All right, Megan and H, if you want to even switch on my microphone, talk to me, that'll be even better. Can get this going faster. So why is using different restriction enzymes an error?
You need like the sticky ends of the plasmid to be complementary to that of the gene fragment.
And you're absolutely correct, right? So therefore, this means that we will not have complementary sticky ends already. All right, okay, so I'll just point for me over here, because then ultimately I will share with you all the answer key, so even easier like this. Okay, so the error here we're going to be then, therefore will result in different and non-complementary. Right, easy peasy, right? And then DNA ligase is missing, so what will happen then? What cannot happen? So think about the function of DNA ligase. If there's no ligase, what cannot happen? Something cannot form. Hey, Megan, does this ring a bell? Without DNA ligase, the phosphodiester bonds will not form. Okay, right? And quite simply, once
Do
we
you need
have
to know
identified, sorry,
do you need to know like the names of the bond?
phosphodiester bond, is it?
Yeah.
Are you asking if you need to know what a phosphodiester bond is?
Or like do you need to write down what type of bond it is?
It is a covalent bond. Is that what you're asking?
No, like the specific name, the phosphodiester bond. Like, do we need to write down that thing?
This you have never seen before. This thing is the phosphodiester bond. If you learn about DNA, the structure of DNA, you should have learned about it. Megan, do you remember? Or do you have your notes to refer to check?
My notes just said the DNA ligase just joins up the fragments to form the recombinant plasmid.
Recombinant plasmid. Okay,
Yeah.
fair enough. Okay. Okay, yeah, actually it's very important for you to let me know as well. So I'm telling you right now that this is called a phosphodiester bond, which joins up nucleotides together. Right. So if you think about it, when DNA ligase from based on Megan's notes is joins up the fragments, right? It is actually like that, isn't it? Right. So then this nucleotide of the plasmid, for example, and this nucleotide of the gene of interest have to bond together, right? So this bond has to be formed. This bond has to be formed, right? So the name of the bond is a phosphodiester bond. But if you say that your school doesn't need you to do so, sure. But why not know exactly what it is, right? Okay, so therefore you will then just say, oh, no, the bonds will not form between the recombinant plasmid. No bonds will form between the plasmid and the gene of interest. Does it make sense, Megan and H now? If you don't say phosphodiester bond, just say, oh, then the bond will not form between the plasmid and gene of interest. Wonderful, thank you to two of you. Okay. Modification is just, okay, use the same restriction enzyme and then add DNA ligase. Okay, fairly straightforward. Sequence of bases in the gene now, right? Sequence is important, right? What the sequence of bases in the gene determines what? Against the sequence of something. Sequence of what? So AUGCCG, no, not AU. ATTCUUG. Why I keep saying U? ATGCCG is different from ACCT But the sequence is very important. Why? This is molecular genetics. Can we keep that in mind?
It will like determine the sequence of amino acids in the polypeptide chain, and then therefore like the protein is like might be different.
That is 80% of what I was looking for. Thank you, Megan. Right? So what are the processors? H, maybe you can jump in over here. The sequence of bases determines the sequence of the amino acids that will be added to the polypeptide during what and what? Do we know the names? Excellent answer, H. Thank you. Transcription and translation. Okay, so that's all. Easy peasy. Let's move on. Okay, so we have a DNA molecule over here, and hence, based on H's two suggestions, transcription and translation, H, which one do you think this is? H, do you think this is transcription or translation? Yes, it is transcription, Megan. Okay, so we are looking for two features in a diagram that supports my answer. Megan, how you know it's transcription?
Because like it only involves the bases. You don't see any amino acids inside.
Uh, Rather than saying what you don't see, maybe you can tell me what you see that tells me that yes, this is transcription. All right, now, Megan. The first one is just, oh, I'm synthesizing an RNA molecule.
I think it's like because you are transcripting the DNA bases into like the RNA bases.
How do you know that? Which base are you looking at?
So for example, you see at the start the T becomes A, C becomes G, C becomes G, A becomes D. So like the complementary base pairings are there.
Complementary base pairing occurs even with the DNA molecules, right? Correct. You see T and A, G and C. So I'm looking for a specific pairing that tells me RNA. What is
Uh,
it?
T and U because thymine and uracil is RNA, so you can see like DNA and RNA like the change.
Correct. Thank you. Right. Okay. So this is our second feature, right? We say that, hey, from the diagram, an RNA strand is made, and the second one is the uracil is complementary to adenine, right? In the DNA nucleotide. Okay, so you've got to keep that in mind. Awesome. Thank you. Fairly straightforward. Okay, come, let's move on. We're looking at the Bengal tiger. Bengal tiger. Okay, so we have the information that the dominant allele for fur color is orange. So orange is the dominant allele, right? How white tigers come about and what is the genotype of white tigers? How white tigers come about? Oh, H, do you mean that two heterozygote parents mate to give rise to a white tiger? Is that what you're saying? Okay, but that's for later. For now, dominant allele for fur color is orange. Why will have white? Why will suddenly have white? That is my clue.
The allele for white fur is recessive.
How did that come about?
Like you have two parents with heterozygous, and then like
Wait. Megan, this reminds me. Just now we were talking about this one. The verb is transcribing, not transcripting.
oh okay here.
Oh, okay. So we're gonna learn how to use that in case next time you're gonna write down the word transcribing, right? The action there. Okay. Right. What else over here? We are looking at how white tigers come about. We are looking at this. The answer for this question cannot be about the cross because the cross is on the next question. So what am I looking for here in this question here, right? What am I looking for in this question here? This one is about, why will have the recessive allele? How did it come about? So original gene is for orange. Why will have white suddenly? Variation. How did that come about? Let's think more smaller picture, not evolution, variation so big. Down to the gene level, the gene itself. What happened to the gene for fur color? What happened to the gene that resulted suddenly got different forms, which is an allele? How did that one gene now have two alleles? Previously, gene is just orange fur. Why now got gene for orange fur and white fur? What must have happened? I'm looking for one word that results in variation. What is that? Yes, mutation. Very good. We're looking for mutation. Okay, right, mutation, right? A mutation of the gene for fur color. Okay, a mutation in the gene for fur color. And the genotype. So this one, the idea here is just how they came about. This one is just mutation. Then what about genotype? What's the genotype for a white tiger here? Based on our dominant being orange. What's the genotype here? Has to be a homozygous recessive, because we're looking at the recessive allele. Okay. So before we look at the genetic cross on the next page here, why are white tigers less common than the orange fur tigers? Why are they less common? There's a link to part A. Why are they less common? Oh yeah, wait. H and Megan, what school are you all from?
So next,
Say Nix, okay. And H. Let's see. Singapore Chinese Girls School. Okay, thank you. All right. Okay, come. So why are they less common? We are thinking about why are homozygous recessive. There's a link to you. There's a clue for you. Why is it less common than the orange fur tigers? So orange fur tigers is what kind of genotypes? What kind of genotypes are here? Orange fur. Megan, come help me, please.
because recessive alleles only express itself only in the presence of another identical allele.
Very good. In other words, they can be what by the dominant? How? Megan, in other words, they can be what by the dominant allele? Suppressed. Okay, suppressed by the dominant, right? So orange fur will be more common because homozygous dominant and heterozygous. Will be expressed as the orange phenotype, while the homozygous will be expressed as the white phenotype, and hence that will be less common. Okay, so learning point for this question is, hey, do you know what a recessive allele is? Okay, and with that, we have come to the fun part, right? We're gonna draw our genetic diagram. All right, question, it has stated using the letter F, dominant allele for fur color. We're gonna show how two orange tigers can have a cub with white fur, and that brings us back to what H mentioned right at the start. Two heterozygote parents. Okay. The more challenging thing about this type of question is actually about the format. Right? We need to be very specific in our headers for our questions here. Right? I hope we can remember that we need to begin with the parental phenotypes. Question only say, yes, two tigers with orange fur. So orange times orange, yes. What are their genotypes? Okay. So this was what H mentioned earlier on. Okay, so let me show you over here. This is what we should be having to see. We're looking at orange fur parents, so parental phenotypes and genotypes. I'm just going to do this split. If your school doesn't allow you to do this split, then repeat parental, right? Parental phenotype, parental genotype, okay? I have a new friend joining us here. Hello, Angeline. Hello, Angeline. And please remember to get the correct worksheet over here. There. Okay, I'll move on. Right. So keeping in mind gametes have to be circled. If in school you all do Punnett square, H and Megan, do you all also do Punnett square? Right. So depends on you, depends on what your school wants you to do as well. So Punnett square or these crosses, both will work, both will bring us to the correct answer. Again, keeping in mind the headers over here are very important. Make sure we have them in a kind of like a neat row, as you can see over here. Right, the rows are very neat and tidy with a cross over here. The gametes must be circled. Even if it's in the Punnett square, gametes must be circled. Okay, and last but not least, I really want to emphasize the importance of having a concluding sentence. Question asks, hey, how can two orange parents have a white kid? You cannot just like, oh, yeah, like that lor. You have to put it down into a coherent sentence, right? So what the parents will do, pass down their recessive allele to the cub. And then, oh, what will the cub have? The homozygous recessive, small f, then it'll be white fur. Okay, so let's keep that in mind now. That's what we're looking for. Okay, so when the exact same cross was done in the zoo, 4 cubs were produced, but the zookeepers did not observe the ratio stated in C. What was the ratio that we saw earlier on, Megan? Do you know what the ratio was? Orange to white.
2, 3.
Sorry, again?
Oh, sorry, 3 to 1.
Very good. So I'm just going to jot this down over here. So 4 cups, right? Look like got 4, right? But the zookeepers there did not see this 3:1 ratio. H, any idea why this is so? Wow. Would you believe it? That's exactly what I'm looking for. Thank you. H, right? So word for word, the sample size is too small. Okay? Sample size is too small. So the observed ratios, which is the thing they actually see, the observed or the actual ratios, will not be seen if the sample size is too small. Okay? Hence, two parts to this answer only, very simple. First part is, oh, sample size too small. Second part is, sample size must be large enough for observed ratios to be close enough to expected or theoretical ratios. Okay? So this part, two things only, right? Sample size too small. Very good, H. Then we want to have need larger for observed ratio to get close to theoretical ratio. Okay. And actually, the next question is about infectious diseases already. Since two of you don't need it, maybe we have the last 10 minutes over here. Maybe I can just directly help you all with something since it's just the two of you. Do you all want to ask me about anything? Maybe other topic also can. You can just shoot your questions now. We can discuss. Let's make good use of these last 10, 12 minutes here. H and Megan, I leave it to you. Come.
Okay,
Whatever you're revising now.
for respiration, right, do you need to know how to differentiate between the internal and external intercostal muscles, the one in the ribs?
Yeah, absolutely correct. You have to. But when you say need to know the difference, is that what you said?
Yeah, because like I saw an MCQ that like had a intercostal muscle and then they asked us to tell whether it's internal or external.
They gave you a diagram, is it?
Yeah.
Okay. So the words internal and external actually tell you what it is already. So if you look at my video now, look at me now, look at my fingers here, right? So my rib cage like that, right? So if I were to open up halfway, you can see the rib cage like this. So the back here is the outside, right? You can see like that outside. So if I were to look at this way, the muscles here are the external, the muscles here are the internal intercostal muscles. Does this help you figure out what the internal versus external intercostal muscles differ? Okay, so because since we are on this topic, maybe you can help me help you to revise this. What happens during exhalation?
When you exhale, first the diaphragm relaxes, so it goes from its flattened position to relaxes into an arched dome position, and then your external intercostal muscles contract, internal intercostal muscles relax. So the movement of the ribs, your ribs and sternum will move downwards and inwards. So the movement of the muscles, ribs and sternum will result in a smaller volume of the thoracic cavity. So this means that the air pressure in the lungs is now greater. So atmospheric pressure outside the lungs is less than the air pressure in the lungs, and then air is forced out.
Absolutely. Megan, can you repeat what you said about the intercostal muscles for exhalation?
For exhalation, external intercostal muscles contract, internal intercostal muscles relax.
Was it
It's opposite.
Wait. External relax. What do you say?
For exhalation, external will contract.
It's relax.
Okay.
Exhalation, external relaxes. Is this? Have you seen this before?
Yes.
Okay, so do you realize your mistake now? You said the other way around, huh?
Okay.
Okay, so ER, external relaxers, internal contract for exhalation. Okay, so don't get too flustered. Just calm down and think about this properly. But other than that, the other explanation points, Megan, wonderful job. Okay, wonderful job. Glad to hear that. Okay, H and even Megan, still anything else that you can want to ask me?
For excretion, right, do we need to know what parts of the nephron and what part of the kidney? Like, for example, do you need to know like the renal medulla contains the collecting ducts?
Yes. Right. So while you will not be specifically tested which part of the nephron is found in which part of the kidney, like the renal cortex versus the renal medulla, right? I have actually seen, rarely, but I have seen questions before they ask, which of these structures, then they point to blah blah blah, which of these structures extends from the renal cortex into the renal medulla. So then the answer to that will be your collecting duct and your loop of Henle, right? These two things will stretch from the cortex to the medulla, and with the collecting duct even going further into the pelvis, the renal pelvis, right? So I have seen it just a handful of times. So therefore, my answer to your question is yes, you will need to know, but not like super important that. Can? Awesome, Megan. And H has a question about ecology. If an insect sucks on zebra's blood, is it that in, huh? H, you want to switch on your microphone and ask me like directly?
Like, if the question states that the insect sucks on the zebra's blood, then they ask you to draw a food chain. Do you draw insect after zebra?
After, then just like that lah. Is that what you're asking? Because H, is that for Hannah?
No.
What's your name? So I just keep calling you H then. Hello, hello. The arrow, you need to understand that it signifies transfer of energy from and to. So as long as the insect obtains energy from the zebra through the blood, this is the food chain, the relationship. Zebra, arrow, insect. Can? And okay. What else? What else?
I have another question under excretion.
Sure.
Okay. So like in hemodialysis, does the speed of the dialysis fluid affect the efficiency of dialysis?
Does the what of the fluid?
The speed of the dialysis fluid affect the efficiency of the dialysis itself?
Is that what is it?
Yeah.
Speed of the fluid. Wow. Okay. Question usually is asking about direction, not about speed. Do you know what I'm talking about? The direction of the fluid. So if we have a tubing this way, and this is my partially permeable membrane, blood enters this way, and the fluid enters this way.
The blood, it comes and goes from the vein, right?
Is it?
Yeah, like sorry, the blood comes from the vein and also goes back into the vein.
Yes,
Okay.
it should. Then that speed thing, basically you must find a balance. It's not a faster, better, or slower, better kind of thing, because both have their benefits. the faster the speed of fluid flow, the faster you can bring out this unit of fluid that has collected bad stuff, the urea from the blood. You can remove it away, right? Okay, that's faster. But then the downside is if this fluid moves too fast, then the efficiency of exchange at each instance will be very low. So then you cannot remove, diffusion cannot occur as efficiently. Then the fluid will not be dirty enough. You will not be able to remove as much dirty substances from the bloodstream. Does that help you understand this speed thing better? Okay, very good. And to your question about the vein thing, right? There are several methods to do it. So the simplest is just, okay, I will draw blood from the vein, I'll clean it, and return it to the vein as well. There's also another method which is taught in schools as well, where they will combine the artery and the vein. Let's say this is the artery, this is the vein. What they will do is they will draw this, they will connect. And then they will draw blood out from here into the dialysis fluid for dialysis. Right, so this is kind of like a halfway thing because vein blood flow will be very, very slow. Then the whole dialysis process of cleaning will take even longer. But I cannot take directly from artery because blood flow will be too fast. So therefore, I find this middle ground. Make sense to you, Megan? So this thing is actually called a fistula. Right, so surgeons will create this fistula first, and then subsequently, dialysis very easy already, straight away from there. Okay. H, anything else? Or Megan? Okay, H is done. Okay, sure. Megan, what about you? Anything else to ask me? Feel free to ask away. Okay, if not, I hope you all don't stay up too late studying for whatever papers you all may have coming up. Well, thanks for spending this 1.5 hours with me then. Hope I have managed to help you all uncover some of the doubts you all may have had for the two topics that we managed to cover here. And as a goodbye, right, once more, my name is Mr. Jason, Mr. Pang. And if you wish to know more about us, we're gonna start all these kind of things soon. And for IP Bio, once you all can have a better communication with me, right, I will be able to know your syllabus schedule better. Each topic I will know lah. I need to know the schedule. If I can match it to your school, maybe you all can grab a few friends together. We can open a class just for your school, and that will definitely be an even better learning experience for you. And as a last thing, maybe you all can help me out here if it's not too much to ask. Fill up this feedback form for me, and once you have scanned it, I don't need you to fill it up at this exact moment. But just scan it first so that next time, once you all have a bit more time on your hands while traveling to school, help me fill this up. But if not, that's all. Thank you once more for spending your Monday night with me. Hope that your first week back to school wasn't too bad. Today's your second week, right? But yes, enjoy the rest of your night, H and Megan. Bye.
Thank you. Wait, is it actually okay if I screenshot some MCQs that I saw?
Sure, which one?
Wait, like
Eh?
not the ones like you showed. Like I've got some MCQs like from my end.
Oh, okay, I get it. Sure. Okay, let me stop the recording first. Hold on.